Hello everyone, and welcome to your Chapter 4 video lecture series where we are going to talk all about cell structure. So we'll start off with section 1 and discuss how we study cells. Our learning objectives are to describe the role of cells in organisms, compare and contrast light microscopy and electron microscopy, and summarize the cell theory. So we'll start off with what the cell is. We've talked about this before, that the cell is the smallest unit of a living thing and really is the building blocks of all organisms. We can talk about a cell or organism as being unicellular, and that means that the organism is just made up of one cell, such as a bacterial cell like E. coli. Or we can say that an organism is multicellular, meaning that we have more than one cell that makes it up. Human being a great example, since we have trillions of cells within our body. And then we can take organisms and classify them as either being prokaryotes or eukaryotes. Prokaryotes, which you can see pictured over here, are going to consist of an organism that does not have a nucleus. So we will have genetic information there, but it will be more within one area, which we call the nucleoid region. Whereas in our eukaryotes, so like plants and other animals, those are going to have a true nucleus that houses our genetic information, our DNA. And we know that tissues can come together and create a tissue. And so these cells will be interconnected in order to have a shared function. And just as a review, tissues can come together to create an organ. Organs come together to create organ systems so that all those organs can have one function or several functions together. And then all the organ systems come together to create an organism. Now let's talk a little bit about microscopy. Now most of us have used at least a microscope at some point in a biology class of some sort, and it is going to be an instrument that magnifies an object. So essentially we could see these smaller cells or materials much more clearly. We're going to use a two-lens system to do this in order to produce an inverted image. And two terms that we really use in microscopy is resolution and magnification. So when we talk about resolution, we're really talking about how clear these images are, these structures are to one another. Sometimes we can have, let's say, two distinct images, and they're so close in alignment with each other that it looks like one thing. But if we have really clear resolution and with enough magnification, meaning that we are like zoomed into that structure, we could see these as two separate structures. And so the resolution will differ depending on the type of microscope that you are using. And we'll talk through just a few of them here, but the one that you most likely have used and possibly will use in the future are compound light microscopes, where the visible light is going to pass and bend through the lens system so that we can see our specimen of interest. When we use lower magnifications, and we can change this up by using what we call the objective lenses here. So for instance, the ones that we have in our microbiology lab, some of them start with a 4x objective lens, where we can basically magnify an object up to 40 times, and then it goes up to 10 times, 40 times, and 100 times, which means we can focus on our specimen up to 1,000 times magnification. So we can see an image of what that would look like over here on the right hand side. We see our red blood cells, and to mention, our cells do vary in size, but a lot of the times we're going to use these microscopes in order to see something that we can't really see with our naked eye, right? And that's how it got its name. Micro means small, scopy means to look at. So if we also use an example such as a pinhead that is 2/1000 of a meter in diameter. So 250 red blood cells could fit atop that pinhead, just to give you something to reference, right? And we can also view live specimens, but we may need some staining since our cells are typically transparent. And when we use these dyes, it typically will kill the cell. So you may be wondering, well, I could see those red blood cells pretty clearly on the image here. That's because we've also used some stains in order to dye the tissues around here. So I mentioned how magnification and resolution are big key terms when it comes to microscopy. So to reiterate, magnification is that process of enlarging an object in appearance, whereas resolution has to do with the revolving power. So our microscope's ability to distinguish two adjacent structures as separate. Remember how we were talking about those two objects looking like one if we do not have good resolution. And you could see some examples in this table down here where we can see the magnification and the resolving power with the different types of microscopes that we have. So when we have higher resolution, we will have better image clarity and detail. And usually scientists will use electron microscopes in order to gain a better understanding of cellular structure and function. On the next slide, let's look at the differences between a transmission electron microscope and a scanning electron microscope. So in general, when we use an electron microscope, we're using a beam of electrons instead of a beam of light, which we see with our compound microscope, so that we can get higher magnification, resolution, and more detail using this electron microscope. When we use a transmission version of it, we are going to see a 2D image. And this really is going to allow us to see more internal structures of a cell. So we don't really have too much going on in this red blood cell here, but if we were to take another cell, we may be able to see a nucleus and a Golgi apparatus and rough ER, and our human eye would not otherwise be able to see that image. So we have to project that onto a screen, and we really can't use live specimens with this. With our scanning electron microscope, we'll see more of a 3D image, and what's cool about this one is that we're able to use it to scan the surface of the cell and really understand the shape and morphology of the cells, and we wouldn't be able to use live specimens here either. Now let's talk about our cell theory. So it was Anthony van Leeuwenhoek who first crafted lenses and observed movements of single-celled organisms, and he called them animalcules. In fact, he would scrape plaque off his teeth and then use these lenses to look at it. And then it was Robert Hooke who then coined the term cell in his 1665 publication Micrographia, when he looked at cork tissue and noticed that there were these chambers which he called a cell. And then we have Matthias and Theodore that proposed the cell theory in the 1830s, essentially having these three things stated as being true: that our cell is the basic unit of life, and that all living things are made up of cells, and a new cell is going to arise only from pre-existing cells. And I just want to reiterate, we can see some of these images of cells on the right hand side. We've got our red blood cells, our osteocytes, which are bone cells within our compact bone, and our nerve cells within nervous tissue. And we can't think of cells as just being those round or square cells. They all are going to have different shapes and really serve different purposes throughout our body.